One of the practical that I'm going to discuss today is the bacteriological examination of water, or we also called it the water quality analysis by a membrane filter technique or membrane filter method. So the membrane filter method or technique was first introduced in the late 1950s as an alternative to the most probable number, or we call it MPN. This is another method for water quality analysis. So to the most MPN procedure for microbiological analysis of water samples. So it involves the use of membrane filters, which are thin porous sheet structures composed of cellulose, esters or similar polymeric materials. They act essentially as two-dimensional screens, and as such all particles, both biological and non-biological, which exceed the pore size are retained upon the surface of the filter from fluids passing through. So bacteria-type membrane filters capable of retaining microorganisms larger than 0.45 micrometer are frequently used for analysis of water. So the objective, what is the objective of this MF technique? So the main objective is to determine the quality of water samples using the membrane filter method. Then the principle of MF technique. So the principle behind is that these membrane filters have a known uniform porosity of predetermined size, generally 0.45 micrometer, sufficiently small to trap the microorganisms. And a water sample is passed through a sterile membrane filter that is housed in a special filter apparatus contained in a suction flask. So following the filtration, the filter disc that contains the trapped microorganisms is aseptically transferred to a sterile petri dish that contains a pad saturated with the appropriate medium. Then the passage of nutrients through the filter during incubation facilitates the growth of organisms in the form of colonies on the upper surface of the membrane. Then the membrane filtration and colony count techniques assume that each bacterium clump of bacteria or particle with bacteria attached will give rise to a single visible colony. So each of these clumps of particles is therefore a colony forming unit, or we also called it in short CFU. So each of these clumps of particles is therefore a CFU, and the results are expressed as colony forming units per unit volume. So the discrete colonies thus formed can be easily transferred to conformation media. So following incubation, the colonies present on the filter are counted with the aid of a microscope, or with the help of a microscope. Then the procedure or the steps involved in this MF technique. So let's see what are the steps or the procedure involved in this technique. So first we will collect the sample and make any necessary dilutions. After that, we will select the appropriate nutrient or culture medium. Then we will dispense the broth into a sterile petri dish, evenly saturating the absorbent pad. Then next we will flame the forceps and remove the membrane from the sterile package. Then we will place the membrane filter into the funnel assembly. So we will flame the pouring lid of the sample container and pour the sample into the funnel. Then we will turn on the vacuum and allow the sample to draw completely through the filter. So after that, we will rinse the funnel with sterile buffered water. We will turn on vacuum and allow the liquid to draw completely through the filter. Then after that, we will flame the forceps again and remove the membrane filter from the funnel. and we will place this membrane filter into the prepared petri dish. And after all this is finished or complete, we will incubate at the proper temperature and for the appropriate time period. Then after that, after we incubate, we will count the colonies under 10 to 15x magnification, and we will confirm the colonies and report the results. So these are the steps involved in the MF technique. So what are the uses and merits of membrane filter technique? So it is used mostly to analyze a series of dilutions of water samples collected upstream and downstream from an outlet of a sewage treatment plant. Then we will have another uses, that is a total count of coliform bacteria will determine the potability of the water source, and the delicate media components that cannot withstand steam sterilization by autoclaving. Example, we have like serum and certain carbohydrate solutions, certain antibiotics and other heat labile substances can be sterilized by membrane filtration. Then the pharmaceutical and cosmetics industries typically focus on monitoring their processed water for Pseudomonas species. So those are the uses. And now coming to the merits of MF technique. So out here, results are available in a shorter period of time. That is one of the merits of MF technique, and larger volumes of samples can be processed. Next, it's because of the high accuracy of this method, the results are readily reproducible, and it allows isolation and enumeration of discrete colonies of bacteria, and it also allows for removal of bacteriostatic and cidal agents that would not be removed in pour plate, spread plate or MPN techniques, which can be done only through this MF technique. So it involves less preparation than many traditional methods and is one of a few methods that will allow the isolation and enumeration of the microorganisms. So we also have limitations of what are the limitations of this MF technique. So during the process of turbid specimens that contain large quantities of suspended materials, particulate matter clogs the pores and inhibits passage of the specific volume of water. Another limitations we have is that when small quantities of sample, for example of sewage, effluent or of grossly polluted surface water, are to be tested, it is necessary to dilute a portion of the sample in sterile diluent to ensure that there is sufficient volume to filter across the entire surface of the membrane. So these are the limitations of MF technique or membrane filter technique. So that's all about the water analysis or water quality analysis by membrane filter technique. Thank you.